Hello, this is the Biology Role Model Project, and my role model will be Karl Deisseroth. So, in his early life, Karl Deisseroth grew up in Boston, Massachusetts. There's not much known about his early life as he's tried to keep a low profile about that, but we do know he attended Harvard for his undergrad and Stanford for his PhD and postdoc. Some obstacles he overcame was that early in his career, the combination of engineering, neuroscience, and physics wasn't really seen as conventional. However, optogenetics, which Deisseroth invented, was met with skepticism as it combined molecular biology, optics, and neuroscience in ways that we haven't seen before. So some hobbies that Dr. Deisseroth have are fly fishing. He says he uses it as an escape from scientific research. And he also likes to write. He has written his own book called Projections, A Story of Human Emotions. And what do I like about Dr. Deisseroth and why I chose him was that he's the pioneer of optogenetics, which is something I want to get into myself in the future. I'm a neuroscience major, and I do want to go down the research track, which means that somewhere down the line I will probably use optogenetics as it's a very easy way to study the brain and very effective. So, neuroscience is the field that he is in. He founded optogenetics, which is a way to use proteins from algae called channel rhodopsins that control neural activity with light, insert them into neurons, and then you can use blue light to activate and inhibit them at the same time. This is a major breakthrough, as now neurons could be studied and brain activity could be studied without damaging or destroying neurons, but instead turning them on and turning them off so that we can keep the patient or different animals alive while we study them, which is great for psychiatry. So one of his recent papers that he was involved with was called Cardiogenic Control of Affective Behavioral State. It studies how cardiac activity influences emotional behavior, and it uses optogenetics inside of the study. So what these researchers did is that they artificially raised the heart rate of rats and then studied how anxiety levels were different in different parts of the brain. So overall, they saw that increased heart rate saw increased anxiety release in most parts of the brain. And they specifically studied one part of the brain called the posterior insular cortex. As we'll see in the next slide, on this panel is some studies they did on this area. So in panel one, I'm sorry about that. It compares time spent in open arms of elevated plus maze between control and experimental groups. No differences observed, indicating that the inhibition of the posterior insular cortex does not affect anxiety-like behavior for this test. In part B, however, it's just showing how lever presses 30% of the time the rats will get a shock, and 30% of the time they'll get a water reward. In C, the experimental group exhibits reduced lever pressing, suggesting lower engagement in the task or increased avoidance behavior when the shocks are introduced. D and E are mirror graphs of each other, and they show the treatment group shows a significantly lower pressing rate, supporting the finding that the posterior cortex inhibition reduces task engagement. This means that integrating cardiac signals with behavioral responses to threats, though its inhibition alone does not lower anxiety as a whole. So a bunch of his major findings are through optogenetics. He's won a lot of rewards for it, which is being studied to use different brain regions all over the world right now. The next step is to move from animal to human trials. A lot of animal trials are being done to study how different brain regions work and how they play a role in certain mental disorders. And finally, we have the reference pages, and thank you for watching.